Hello students, welcome to the EPG Pathshala. I am Anil Kumar Sharma from Department of Pharmaceutics, Delhi Institute of Pharmaceutical Sciences and Research, affiliated to University of Delhi. Today, I am going to talk about module Nanoerythrosomes from paper Novel Drug Delivery System 2. The learning objective of today's molecules are 1. Erythrocytes and their role in drug delivery, how they can be used in drug delivery. Second one is nanoerythrosomes, what they are. Third one is prerequisite for drug molecules. What are the characteristics a drug molecule should possess to be used, to be entrapped in RBCs. The limitations, the isolation techniques for red cells, and lastly, storage of RBCs as well as nanoerythrosomes. The erythrocytes. What erythrocytes are? Erythrocytes, the name has been derived from erythro and cytes, is the portmanteau of two words, erythro and the cyte. Erythro means red and the cyte refers to the cell. So that means erythrocytes are red blood cells. Morphology of red blood cells. The cells are oval, biconcave and without nucleus as well as most of the organelles to accommodate hemoglobin. The hemoglobin is concentrated in the RBC. Normally, these RBCs are on an average 5.7 micron in diameter and around 2.5 micron thick at the thickest point and 1 micron or less at some thinner points. The number of RBCs also vary. The average number of RBCs in a healthy man is around 5.2 million and in case of women it is 4.7 million. The people who are residing at higher altitudes may be having more number of RBCs. To accommodate for the need of their oxygen, they have to generate more number of RBCs. Now we will come to the lifespan or the life duration of the red blood cells. Normally, the cell's life is around 100 to 120 days in normal humans, and after 120 days your RBCs are going to expire. But in case of Wistar rates, the span of RBCs around 60 days. This figure shows the morphology of erythrocytes. You can see they are biconcave or the oval disc, biconcave at the center, and the dimensions, as which I have told you earlier, are similar to that one. Erythrocytes. The erythrocyte membrane is made up of cholesterol and phospholipids and has an elastic scaffold of skeletal proteins via transmembrane proteins. Membrane is bilayer in nature and makeup of outer and inner layer is as per requirement. Owing to membrane fluidity and flexibility, erythrocytes may squeeze themselves across much narrower opening than their diameter and can recover swiftly to their original shape. Phosphatidylcholine renders highly fluid lipid regions to induce fluidity, while sphingomyelin contributes towards rigidity of membrane. Lipid domain regulating the mobility is determined by molar ratio of cholesterol to phospholipid, extent of unsaturation of phospholipid acyl chains, and phosphatidylcholine sphingomyelin ratio. A vital factor in RBC vitality is its flexibility. Main variables influencing deformability are inherent viscosity, the surface volume of the cell, and intrinsic flexibility of the membrane. The RBCs possess other very fascinating features like those that they serve as an osmometer because they shrink upon placing into a hypertonic solution or swell while kept into a hypotonic solution. The RBCs may attain a crucial hemolytic volume, producing perforations over the membrane in the range of 10 to 500 nanometers. These procedures are generally reversible After hemolysis, the perforations close and the cell requires its biconcave shape. Production of new RBCs. All the RBCs or all the blood cells are being formed into bone marrow from a single cell that is known as hemocytoblast. And we will talk in particular with the RBC only that are converted from proerythroblast to polychromatic erythroblast. Then erythrocytes are resulted. Advantage of erythrocytes as drug carrier. The RBCs offer biocompatibility as autologous cells are being utilized, thereby eliminating the chances of triggering immune response. Other, biodegradability. The biodegradability as there is no production of toxic products. Uniformity of size. Since all the cells are from same population, they are small in size and uniform in size as well. RBC's environment is inert because this is devoid of cell nucleus and most of the cell organelles. Protection of loaded drug through inactivation by endogenous chemicals. The broad classes of the drugs may be encapsulated into the RBCs. Improvement of drugs pharmacokinetic and pharmacodynamic profile. As the RBCs are offering this improvement in the drug's kinetic and the pharmacodynamic properties or the variables. Other thing that RBCs offer steady state plasma concentration that is without fluctuation and that makes the patient compliant towards taking the drugs. These offer safety of patient versus the toxicity caused by some of the cytotoxic drugs like antineoplastics. The RBCs makes the drugs enable to detour immunological response as the entrapped drugs also bypass the other drug because of autology of the RBCs. Scope of technology improvement. There is a scope to handle the drug segregating, transfusion and dealing with erythrocyte. It offers to for the scope or the room for the advancement. Targeting to reticuloendothelial system organs. The RBCs are having potential to target the organs which are of RES system. The RBCs offer zero order release of actives. That means a constant fraction of drug is released irrespective of the initial drug concentration. Further, the RBCs offer higher drug encapsulation. The drug entrapment is to the higher side because of small size encapsulating the higher volume of the drugs. They offer longer circulation period. As the circulation of the RBC is they travel lots of miles in the blood circulation system, that's why offering longer circulation period. Further, these are offering advantage in respect of patient compliance. They are reducing the dosing frequency of the drugs being administered. Now we will talk of nanoerythrosomes. Nanoerythrosomes we defined they are the tiny vesicles made by the sequential extrusion of RBCs through polycarbonate membrane filter of varying porosities under the influence of nitrogen pressure. These are having an average diameter of 100 to 200 nanometers. That's why these are termed as nanoerythrosomes because the size is lying in the nano range. Morphology involves this, it is a regular vesicles in shape and the morphology involves phospholipids, proteins in the structure. Features. The liposomes or nanoerythrosomes are the vesicles made from the RBCs and a single RBC can result into 4000 to 6000 nanoerythrosomes. The figures shows a fluorescence image of nanoerythrosomes which are tiny vesicles. They are being used, they are buoyant vesicles and being used for number of drug delivery applications involving targeted drug delivery, sustained drug delivery of biopharmaceuticals as well as the large molecules like proteins and peptides and the ribonucleases. So in future they are finding to deliver the biopharmaceuticals as well. The biosimilars are now drugs which can treat number of diseases which are occurring today. Composition of nanoerythrosomes cell membrane. Their membrane is made up of about 50% phospholipids, 25% protein, and 25% cholesterol. This characteristic is of paramount significance upon comparison of such particles to other nanoparticle delivery systems like liposomes, as it permits a high degree of versatility in the type of molecules that can be entrapped within the nanoerythrosomes. The molecules like carbohydrates, proteins, DNA, chromophores, and the many drugs. Besides, it eases the covalent coupling of different molecules to the nanoerythrosome surface, which includes receptor ligands, antibodies, anchoring proteins, polyethylene glycols. Further, passive targeting may be more influential by availing benefits of difference between normal and tumor vasculature. The applications of nanoerythrosomes. These cells could be employed as circulating carriers to distribute a drug for long duration in circulation or in target specific organs such as liver, spleen, and lymph nodes. Numerous drug delivery research employing drug containing erythrocytes are under clinical trials. Antineoplastic drugs like methotrexate, bleomycin, asparginase, and adriamycin have been formulated using erythrocytes as carriers. These cells offer elimination of RES iron high amount, exit of toxic substances, and delivery of antiviral agents, and few other uses of resealed erythrocytes. These cells offer delivery of biopharmaceuticals, therapeutically important peptides and proteins, nucleic acid based biologicals, antigens, and vaccines, are contributing to the latest core pharmaceuticals for drug delivery exploiting carrier erythrocytes. Drug molecule and the RBCs. What are the various characteristics which a drug molecule must possess to be encapsulated into the RBCs? A drug candidate should possess high water solubility. The drug molecule must possess great extent of hydrophilic characteristics, although some hydrophobic drugs are also delivered using cellular carriers. Resistance towards degradation within erythrocytes. The drug moiety should be robust enough to withstand the chemical degradation in erythrocyte environment. Thus, drug is also keeping pace with the long circulation period of cells. Non-toxic to erythrocyte membrane. The drug must be innocuous to the surface of cells so that the integrity of membrane is maintained. The alteration in membrane composition may lead to failure of this delivery system. Establish pharmacokinetic and pharmacodynamic features. The drug molecule should have an obvious profile and defined targets in order to facilitate its compatibility with erythrocytes. Although erythrocytes offer many advantages over other drug carriers, but they also suffer from some constraints as listed below. Elimination by reticuloendothelial system shortens circulation time. The erythrocytes are taken up by RES cells where degradation takes place, causing the former to release its contents. This eventually leads to incomplete drug delivery. Drug leaching. Sometimes the entrapped drug may come out of vesicular carrier, which may give a subtherapeutic concentration at site of action. Some drugs cause morphology alteration and that have deleterious impact on cell membrane of erythrocytes, thereby altering the surface features. There may be individual variations due to diversity in features. This may be encountered in certain cells due to genetic setup changes. Isolation of red blood cells. Isolation involves blood withdrawal. Blood withdrawal from any of the mammal species like human, rat, rabbit, dog, mice, or any of the animals which can be used for blood withdrawal. Blood can be withdrawn by using aseptic techniques. These aseptic techniques, that means environment should be sterile. The vials in which we are collecting or the tubes which we are collecting should be free from any pathogen or the pyrogen or anything. The blood is collected in vials containing a drop of anticoagulant. The anticoagulant normally is used EDTA. Since EDTA, what EDTA is? EDTA is ethylene diamine tetraacetic acid. This is most commonly used anticoagulant because it best protects the inherent characteristic of red blood cells. Other than this anticoagulant, heparin or the many more anticoagulants are being used. How we collect the blood? We collect the blood in vials containing anticoagulant, as I uttered. This anticoagulant containing vials which we have collected the blood. Now centrifuge to collect packed RBCs. This packed RBC is the term we use while transfusion. Every time we use packed RBC and the parameter known as hematocrit value. These RBCs are now subjected to number of washing, or we call them repeated washing. We can wash them one by one, other next and next and next to obtain a pure dispersion of the RBCs. The hematocrit that we commonly use is in the range of 5 to 95%. But normally we use or we work with 70% of hematocrit value. That is very quite easy to work with. Isolation of RBCs. Briefly, the procedure includes blood withdrawal from small animals as well as large animals. In case of small animals, blood is taken from by cardiac or splenic puncture. In case of large animals, it may be drawn by venipuncture. The blood is collected into heparinized tubes by venipuncture. The whole blood is centrifuged at 2500 rpm for 5 minutes at 4 degrees Celsius in a refrigerated centrifuge. The serum coats are carefully removed and packed cell washed 3 times with phosphate buffer saline of pH 7.4. Washing of packed cells 3 times with phosphate buffer saline of pH 7.4, then dilution and storage at 4 degrees Celsius. Then these cells are resuspended in buffer solutions, and afterwards these cells are stored in acid citrate dextrose buffer at 4 degrees Celsius up to 48 hours prior to its use. The anticoagulant used for isolation of RBC is composed of, per 10 ml it contains lithium, potassium or sodium oxalate in the range of 15 to 25 milligrams, sodium citrate 40 to 60 milligram, heparin sodium 2 milligram, disodium or tripotassium ethylene diamine tetraacetate in the range of 10 to 30 milligrams. Lastly, it contains acid citrate dextrose formula B solution amounting to 1 milligram. The shelf life of RBC and the nanoerythrosomes. These may be prolonged through storage in a refrigerator using preservative solution. Majority of preservative solutions facilitate storage of about 42 days consecutive to collection. For instance, saline adenine glucose mannitol is used as preservative solution. The time duration of 42 days as expiry of RBCs is derived from yardsticks established by United States of America Food and Drug Administration, which states that 75% of transfused RBCs ought to be retrievable in peripheral blood circulation 24 hours following transfusion. Alternatively, the RBCs may be stored as frozen product, which involves use of glycerol in the concentration of 40%. This is followed by storage at minus 65 degrees Celsius for 10 years. That's how we can increase the shelf life of your RBCs by storing them at as a freeze dry products. While used, the frozen product is first thawed at 37 degrees Celsius and deglycerized prior to transfusion. In general, usual storage media for nanoerythrosomes is Hank's balanced salt solution and acid citrate dextrose at 4 degrees Celsius. There are some interesting facts, students. Erythrocytes have to travel about 70,000 miles every 20 seconds. You see, we don't think of it while the RBCs are traveling such a longer distances in such a small time of 20 seconds. Every day, 200 billion RBCs are produced. See the large number. Iron is maximum in hemoglobin. As we have seen earlier, the RBCs are having the ability to concentrate hemoglobin to the range of 34 gram per 100 ml of RBCs. The iron needs of RBCs are fulfilled from dead RBCs. So that is the utilization of the dead RBCs in the cost effective manner by the body itself. Body is having the mechanism to use the iron from the dead RBCs itself. We continue to tell you about the interesting facts about the RBCs associated with the nanoerythrosomes. The person who reside at higher altitude have more number of RBCs. You see, sometimes people are having anemia. They are suggested to go to the higher altitudes to raise the number of their RBCs because they have to be in a position to cope up with the deficit of oxygen at higher altitudes. The RBCs were firstly discussed by John Swammerdam in 1958. So that person is the pioneer in discovery of these cells. Do you know these crocodile ice fishes do not have RBCs? The red color of RBC is because of hemoglobin. Other than mammals, there are salamanders or the other species which don't have the nucleus into their RBCs. The RBCs used for blood transfusions are known as packed RBCs. The RBCs are evaluated on the basis of hematocrit value. The generation of RBCs is called as erythropoiesis. That means generation of new RBCs. The RBCs are born in bone marrow and mature there only. Ethylene diamine tetraacetate is generally used anticoagulant which protects the red cells from harmful action. Although number of anticoagulants are being used for as anticoagulant which are protecting the RBCs from harmful action, then the capillary hollow fiber plasma separator is more efficient in washing of red cells. We use normally buffers to wash the RBCs. Usually the hematocrit value of 70% is easy to work with. Normally we take the hematocrit value of 5% to 95% in case of RBCs. Any percentage based on the requirement can be used for the treatment or for the nanoerythrosome preparation. Approximately 4,000 to 6,000 nanoerythrosomes can be made from a single RBC. You see, one RBC can yield you around 4,000 to 6,000 nanovesicles, which is cost effective too because the loss of RBC is much less as compared to using RBC as drug carriers. The nanoerythrosomes have larger surface area to volume ratio than red cells themselves because they are smaller in size, having a diameter of 100 nanometer or lesser in size. The circulation time of nanoerythrosomes can be increased by surface modifications. These modifications include the coating with polyethylene glycols or the other molecules of other functional groups we can add on the surface of nanoerythrosomes membrane, thereby increasing the residence time into the circulation. Eventually, it leads to improvement in the drug delivery profile of the drugs themselves. So, students, let us now summarize what we have learnt in this module. We have to make a precis or the summary of whatever we have learnt in this module. First, we have gone through the morphology or the definition or the characters of erythrocytes. These erythrocytes, how these can be used in drug delivery purposes. These erythrocytes are the vesicles without immunogenicity. They are biocompatible, they are biodegradable. So these can be used as drug carrier because of their own virtues, own inherent character. Because autologous cells are used, there is no chance of triggering immune response. Along with that, their circulation time is much more as compared to other carriers or the other vesicles. So we can use erythrocyte as a potential drug carrier in the drug delivery. Now we will come to the nanoerythrosome. Nanoerythrosomes are the vesicles generated from the erythrocyte ghosts. First, the erythrocyte ghosts are being made. These ghosts are transformed into the nanoerythrosomes by number of techniques like extrusion, sonication, or the electroporation. And nanoerythrosomes are made in cost-effective manner too. One erythrocyte can result into 4,000 to 6,000 nanoerythrosomes. So number of erythrocytes we are going to use are somewhat lesser, but nanoerythrosomes we can make number of nanoerythrosomes by using single erythrocyte. So that is also helping us to make a drug delivery system that is cost effective. That means we are making the drug delivery system in economic manner and we are helping the mankind in a good way. But there are some limitations of these systems also. There are offering number of advantages, but limitations are the technique is expensive, chances of drug leachings are there. Some drugs may cause deleterious effect on the membrane of cell. These all lead to the failure of the delivery or the improper delivery leading to dosage regimen which are of subtherapeutic level. Then we discussed isolation of RBCs from various donors, various techniques to isolate RBCs, how the RBCs are being isolated. The isolations and their storage of RBCs as well as nanoerythrosomes. The storage of RBCs and nanoerythrosomes is normally containing preservatives. The saline, adenine, uh, solutions are being used. Normally acid citrate dextrose solution is being used or Hanks balanced medium is being used for storage of nanoerythrosomes. So these carriers being biodegradable, biocompatible appears a promising carrier, promising vehicle for drug delivery for number of active pharmaceutical ingredients. The single RBC is giving you 4,000 to 6,000 nanoerythrosomes. These nanoerythrosomes are being used in a variety of drugs to deliver into the biological system. They are helping the mankind in a good manner. A number of drugs meeting essential features. That means there are certain characteristics which a drug should meet in order to be encapsulated into the RBC. So those are meeting the requirement, they are being encapsulated and delivered by using RBCs. Thank you very much.